Well, hello, everyone. I'm Brian Culley, CEO of Lineage Cell Therapeutics, and I'm really happy to be able to share our story today at the third annual SCI Investor Symposium. As Lineage is a publicly traded company, I'll refer you first to our safe harbor clause in case I make any forward-looking statements today, and all of this information is available through our filings with the SEC.gov. So the lineage approach is that in certain settings, we believe, and we believe there's evidence that replacing whole cells may provide restorative benefits that are simply beyond the reach of traditional approaches. So the traditional approaches, of course, are small molecules and antibodies, but in some places where whole areas of cells have become destroyed or dysfunctional, replacing those cells may be the only way to restore function and activity. Lineage has a pipeline of cell transplant approaches. The most notable one to date is our retina program, which was partnered in a $670 million deal with Roche and Genentech. But today I'll only be talking about our OPC1 program in spinal cord injury. So the approach that we use to aspire to regain function in individuals who've suffered from a spinal cord injury is really just comprised of two steps. So we begin with a cell line of pluripotent stem cells, and these cells have this wonderful property that they are self-renewing. So the cells will continue to replicate, giving us essentially an endless supply of cells. The other property that these cells have is that they can become any of the cells of the human body, including, of course, cells that comprise the spinal cord. So we put those undifferentiated cells through a process, through a differentiation protocol, which guides them into becoming just the cell type that we want. And in this case, that cell type is the oligodendrocyte progenitor. We do not put stem cells into patients. The stem cells always get converted into one of the glial cells, and the target cell type, of course, is the oligodendrocyte progenitor. Importantly, if you want to have a successful cell therapy, you need to have a lot of properties of your material. It has to be well controlled for safety. It has to be reproducible, so it's the same, the identity, the potency, and of course the scalability if you want it to actually get to the patient population that you're developing it for. This is a lot harder than it looks, but we think we've solved these problems, in particular with our spinal cord program. One of the recent accomplishments that we've been able to share publicly is the manufacturing capacity, the manufacturing scale we have for these cells. Importantly, this is an off-the-shelf solution. So we're not taking cells from a patient and converting them or manipulating them and transplanting them back in. That's a customized therapy, which typically is tremendously expensive. What we are doing is we have a cell line that is the population for all of the therapies that we make for patients. So just one cell line can create a master cell bank, which then any of those vials of the master bank can create a working cell bank. And then from there, you can make your product. So if we wanted to manufacture enough material for the 18,000 people every year who suffer from an injury in the United States, we would just run this process multiple times. And that's an important accomplishment because therapies, increasingly, it's imperative that they're affordable and accessible. So if it costs you hundreds of thousands of dollars to make your material, which it does not cost like that for us, it really becomes a challenge to the system. So we are trying to solve this by having one product that's fit for all. All right, so our product candidate is called OPC1. These are, again, oligodendrocyte cells, and they are transplanted directly to the spinal cord, to the area of injury. There are 30 courageous and fantastic individuals who have participated on our clinical trials to date, and there's been some really exciting results that we've seen. The reason that we are approaching the condition in this way is that we're aware that there are a lot of technologies that can, extend axon length or grow dendrites, but one of the things that's absent is the critical myelin, the the insulation that wraps around these cells, and that's provided by the oligodendrocyte. So our approach is to provide the oligodendrocytes, which permit the neurons to function normally. They need that wiring to be connected and in order to fire in a cohesive and non-spastic way. So we believe that transplanting oligodendrocyte cells directly to the site of injury could provide the kind of solution that is needed, or at least part of the solution. Again, these cells are allogeneic. This is a formulation that we've been working with that's off the shelf, and it's a one-time treatment. To date, everyone who has received this therapy has received it 3 to 6 weeks following their injury. But as I'll explain in a little bit, we're expanding and beginning to look at individuals who have chronic injuries between 1 and 5 years old. Not the individuals being 1 to 5 years, the injury being 1 to 5 years. This does not require lifetime immunosuppression. We have very long follow-up, up to 10 years on people who've been in the trials. The immunosuppression schedule is just 60 days, and it's, again, a very convenient thaw and inject formulation that we've developed for our cell manufacturing system. So how do these cells work? Well, there might be multiple mechanisms of action. We're very excited, certainly, that they can prevent the cavitation. This is a rodent spinal cord slice. This is not from a human, but you can see going from left to right in the upper panel that after transplant of the material, there's a very visual normalization that occurs. Obviously, electrical impulses cannot jump gaps or vacancies. You need some material, some conductive material present, extending that tissue bridge could be beneficial. Myelating the axons, right? Again, with that material that's normally and naturally found there, we think that might be critical to an effective therapy. And these cells are known to secrete neurotrophic factors. We don't know what percent each of these functions may serve in the overall effects that we've seen to date. However, it may be that they're working in collaboration for best possible outcomes. The clinical data to date has been evocative. We do know that in this population, there often is spontaneous recovery to different degrees, and it's very hard to find perfectly matched groups. But when we've made our best effort to try to find a suitably matched patient population and compare it with the 25 individuals who had a cervical injury in the last trial, we think that there are benefits here. We find this through conversations with the PIs. We find this for conversations with the individuals who went through the transplant and have enjoyed some benefit. And we think it compares nicely. But of course, ultimately, the true test of this would be in some sort of controlled study with some randomization feature. But certainly the phase 1 data where 96% of people enjoyed some improvement and no one had a loss, we think that that's promising and merits further investigation. For those individuals who did gain 1 or 2 levels of improvement, these can be tremendously beneficial increases in function. My experience with patients with lived experience is that they've taught me that even very small gains can be very meaningful. I think it's important that we entrust the individuals with the injuries to tell us how important the gains are and what kind of gains matter. But we do know from research that 1 or 2 levels of gain can massively reduce the dependency and the reliance on the supporting caretakers and caregivers. And that provides not just an economic benefit, but even a huge social benefit. The challenge of getting all of that care and having it consistent, reliable, is a tremendous unmet need in this population. So we're really excited that about a third of people who were on our trial did in fact see 2 levels of improvement on at least one side, which could really improve their quality of life. Even more importantly, the safety phenomenon of these therapies must be closely noted. In this case, among the 25 patients, there were 534 adverse events. Please keep in mind, many of these are vehicular accidents or other severe injuries, so there are a lot of complications. You would expect that. But out of those 534 adverse events, there was only one that was potentially related to our treatment. That was a grade 2 dysesthesia, which did self-resolve. So we think the safety profile here is really compelling. Another note out of the last trial that was conducted was the capacity for OPC1 to prevent the syringomyelia, which can occur, filling in that vacancy, forming a nice tissue matrix that's durable. 96% of individuals had no sign of a lesion cavity at 24 months. That's among the 22 available scans that we had. So we see this interesting and encouraging safety profile. We see this interesting and encouraging efficacy signal, and we see these additional signs of benefit through areas such as syringomyelia. These trials that we've conducted, both the phase 1 in 5 thoracic patients, as well as the phase 1/2A in 25 cervical patients, they've all been recently published just a few years ago, and it's really exciting. You can go into tremendous detail about how these patients have performed, and there in fact is additional publications that will be coming out from this work. Most notably, I know that there's an MRI-focused publication that's forthcoming. One of the things that Lineage is tremendously proud of and quite good at is controlling the manufacturing of cells. And we do know that in these early studies, the cell populations were not really at the optimal level, or at least the level that we wanted. So when we acquired this program, we invested in changing how the cells were made to try to purify and really control the process better. And this image shows you some single-cell RNA-seq data comparing the original clinical material that generated the data I just showed, compared with the material that we have improved in the process we have. And if you look in the top left-hand corner of the original material, we've shared with you, a lot of this is confidential, but we've shared with you that population of purple cells, that population are epithelial characteristics, identity, and we've been able to eliminate them. We don't think they're suitable. We don't think they're necessary or helpful. And so we think that the new tools of cell therapy, including the bioinformatics and single-cell RNA-seq, really help companies like ours to understand, are we manufacturing the right material in a consistent and predictable way? Do we have good control? And oh, by the way, we increased the scale more than tenfold when we are doing this. So that helps with the production costs to lower those to create a much more attractive manufacturing capability. In addition to the cell manufacturing, we also are working on the delivery. The old way of delivering these cells required a big scaffold, this apparatus that held the needle. It really is not the kind of thing that you want to take past phase 1. I mean, it's fine for phase 1, it worked. But if you want to go out and do larger studies at dozens of sites, or you certainly want to ultimately have your technology available to a very large, many thousands of individuals, you really have to have a simpler system. So we're right now running a clinical trial where we're looking at this device on the right, which is directly attached to the patient. So it's not attached to the bed and have this overhead scaffold. It's directly attached to the patient. And the reason why that's important is that while the patient is breathing, the whole apparatus is moving, the flow path of the cells are moving, the needle's moving. That means that you can actually deliver our therapy very slowly to the spinal cord over 4 or 5 minutes. That was not the case in the original trials. You actually had to disconnect the patient from the respirator, and very quickly, within a minute or a little bit more, you'd need to deliver the therapy. So in our case, you don't have to stop the patient ventilation, which we think is safer. And of course, this device is a lot simpler and easier to use. It also gets rid of the dose preparation from the new formulation that we are introducing next into the clinic, which is much easier to use, doesn't require hours of preparation beforehand. It's a very quick thaw and inject formulation. We're excited to get this new device tested and then get the new formulation tested right afterwards. This new study has only just opened at the first site at UC San Diego Health. It will aim to treat 3 to 5 subacute patients, and for the first time ever, 3 to 5 chronic injury patients. It's really exciting because we've never seen what happens when our OPC1 cells are administered to someone with a chronic injury. So we will be following not just, of course, the safety and the performance of the device, but we'll be looking at other assessments of functional ability over longer periods of time, because we'd love to know if these cells do change someone's trajectory in the out years. that's less common, and it would be an interesting and evocative idea for this therapy. But ultimately, to be very clear, this study in 6 to 10 patients is really aimed at assessing the safety and performance of this new delivery system that we have so that we can employ it and deploy it into larger comparative studies of OPC1. So, summing up here, one of the great things that we're really proud of in the OPC1 program, we acquired it a few years ago. We knew it had some areas of improvement, but it offers us unmatched experience. This is one of the longest running trials. We have very long safety data. We see these exciting signals that there may be some benefits from people who have received these therapies. We have greatly improved the production and the scale of the material, and we learned a lot in those first studies. So now we're moving on and solving some additional problems around delivery. We're preparing for larger controlled studies and asking the right questions about how to design and conduct those studies, working closely. We will be working closely with FDA on what that looks like. And we have really benefited wonderfully years ago from funding from the California Institute for Regenerative Medicine, and we are seeking additional support for this trial from that valued partner. So the OPC1 overview is this is using a targeted cell replacement. It's a similar approach to the success we've had with RPE and dry age-related macular degeneration, or you may see some of the great news coming out of the Parkinson's field or diabetes. In short, people are using differentiated and specific cells to show what really appears to be functional cures in massive unmet needs. So this is a really exciting new branch of medicine, and we're so proud to be pioneering a segment of it. The OPC1 program has RMAT and orphan drug designation. It's received this wonderful support from CIRM, and it may have application in other demyelinating conditions. So if you want to learn more about this program and our work at Lineage Cell Therapeutics, we have a lot of content available on our website. So I really thank you. Please do consider coming to visit us either in Carlsbad, California, or we have a manufacturing facility in Israel. Company is squared away and financially suitable to be able to reach some important milestones. And, I think this year we'll probably have some updates on the recently opened clinical study. So thanks for your time and interest, and, please continue to follow our story. Thank you again.